Hello friends! In this video, I'll talk about the animal cell culture. This video is important for those who want to set up animal cell culture lab. I'll give you the brief introduction about animal cell culture, what are the equipment you need to set up the lab, as well as you will find a layout of the lab in this video. Also, we will learn about how to perform the primary and cell line culture with examples. So keep this video at the end, subscribe and like my channel. Let's have a look on brief introduction regarding animal cell culture. Cell culture is the process by which cells are allowed to grow in controlled conditions outside of its native environment. As we know, the cells grows and dies continuously inside our body. In this technique, we can grow particular type of cells outside the body in controlled conditions. So it is defined as the isolation of cells from animal and then grown in vitro in culture media aseptically. For example, have a look the diagram. Lung cells has been taken out from the organ lung and kept in its favorable media and conditions. So lung epithelial cells can be cultured in this situation. This technique has enabled researchers and scientists to use cells as an imperative model system for understanding the physiological processes in the human body. Now let's talk about brief history of animal cell culture. One of the major contributions of cell culturing is the developing the polio vaccines. In the 1930s, researchers had to use animals to grow polio viruses, but after developing cell culture techniques, scientists were able to have great control of virus production, which eventually led to the development of the vaccine. Fast forward to 1970s, mammalian cell culture was used to create properly folded and glycosylated proteins such as interferons and antibodies that were impossible to make in prokaryotic E. coli cells because they lack post-translational modifications. Nowadays, cell culture is being used widely in lab settings, particularly in the vaccine research, genetic engineering, gene therapy, genetic counseling, and many more. Coming to the next, cell categories. Cells that are used in laboratories can be generalized into two categories. First is primary cells and another cell lines. Now first we learn about primary cell culture. A primary cell culture is prepared by inoculating cells directly from the tissues of an organism into culture media. For example, lung tissues can be isolated from the organ directly and disintegrated manually or biochemically by using pipettes up and down. Once the cells disintegrated fully, we plated the cell suspension into its favorable media and allowed to grow aseptically. After some weeks, we can see the layer of cuboidal epithelial lung cells bottom of the plate. Now coming to the types of primary cells. They can be divided into two types, adherent cells and non-adherent cells. For adherent cells, they need a surface to which they can attach to grow. Thus, they also called anchorage-dependent cells. Example, cells normally derived from organs. Non-adherent cells, they do not require any surface to grow. Thus, they called non-anchorage-dependent cells. Example, lymphocytes. Generally, the plate need to be coated with extracellular proteins like laminin and fibronectin so that these types of cells get attached at the bottom of plate easily and grow. Although non-adherent or suspension cells do not need any such type of substrate to grow, they float in a medium and many requires agitation for adequate gas exchange. These primary cells has different growth kinetics. First of all, there would be a lag phase, which is an initial phase where cells prepare itself to adapt the environment and there is no significant growth take place. And then followed by a log phase where maximum cell growth takes place in exponential fashion. After some times, due to depletion of media and nutrients or lack of space for new cell growth, and accumulation of waste product will lead to plateau phase. In this phase, cells will become confluents and they need to passage in different hubs. Primary cell culture usually heterogeneous and slow growing, but are more representative of the tissues of their origin both in cell types and properties. So once the primary cell become confluent, they need to subculture into other hubs because newly growing cell didn't find a space to grow more. So this process gives rise to cell line. Now let's talk about cell lines. Cell lines could be finite or it could be infinite or continuous cell line. Finite cell line which may die after the several subculture and have limited lifespan. This is because each time as the cells divide, parts of its DNA's telomere is lost and after a certain number of cell division, the cells would reach on its senescence stage when the cells can no longer divide. Infinite or continuous cell line may continue to grow indefinite or has an unlimited lifespan. Usually, normal tissues give rise to finite cell lines, while tumor give rise to continuous cell line. Coming to the established or continuous cell line, mostly normal cells survive in culture for a limited period of time ranging from 20 to 100 cell generations. Usually, normal cell lines will grow for about 8 to 10 subcultures or about 12 weeks, after which they go through a crisis and cease to proliferate. If a culture survives this crisis, most of its cells die out, while some cells proliferate to give rise to continuous cell lines. The phenomenon that generates continuous cell line is called in vitro transformation, which may either occur spontaneously or may be chemically or virally induced. The word transformation is used to describe this phenomenon because continuous cell lines have marked similarities with malignant cells and are often tumorigenic. The reagent or treatments which are responsible for in vitro transformation can target one of the checkpoints in the 5 phases of mammalian cell cycle. Checkpoints are there to ensure that the cells meet the appropriate requirements in order to move on the next stage of making daughter cells. One of the checkpoints at the end of G1, which allows cells to commit to cell division. The second checkpoint is at S phase and is there to check the quality of DNA and to determine if any DNA repair mechanisms need to be activated. The third checkpoint is after G2 and its purpose again to ensure that the DNA has been replicated completely and is undamaged. Finally, there are several checkpoints within mitosis to ensure that the cell is in a proper position to complete cell division. When a cell starts to divide uncontrollably, it is because they have evaded these checkpoints and the cell cycle is allowed to proceed and cells continues to proliferate. Many viral oncogenes or tumor suppressor genes such as E6 and E7 from the human papilloma virus can degrade controllers of the cell cycle, which turn a normal cell to cancerous. Coming to the next how to perform primary culture, but before to continue, we should learn the list of some major and minor lab equipment we need to perform the experiment. So what equipment do we need to set up a cell culture lab? Let's have a look. So most important, laminar air flow with biosafety level 2. All the work should be done inside the hood to avoid the contamination. First wipe the hood with disinfectant and arrange all the materials inside the hood before starting the experiment. And we need the CO2 incubator to keep the cells inside for optimum growth conditions. Refrigerated centrifuges need to pellet down the cells from the suspension. We also need a good phase contrast microscope to visualize the cells. Of course, we need a cell counter to check the viability of the cell. Minor equipments include autoclave machine, micropipettes, and media aspirator. And of course, all plastic wares which will be used to do a cell culture. So have a look at generalized layout of cell culture lab. All the major equipment should be placed in a closed, fully air conditioned room Before you enter the lab, maintain the hygiene, like remove shoes, wash your hands, wear lab coat, and then enter in the lab. Autoclave should be placed in outside the culture room where you can prepare the media and packed it and then sterilize it in the autoclave machine. Let's have a look now how to perform primary cell culture. I am taking an example of normal ovarian surface epithelial cells. So first ovarian tissue is taken out from the patient. Make sure you have to take consent from the patient before taking the sample. From any patient surgery, the surgical staff can give you the sample in sterile container containing saline. So take out the sample in Petri plate and add the space tube for disintegration the tissue and incubate it for 30 minutes. Of course, all this work should be done in tissue culture laminar flow hood. After that, add pre-warmed suitable media like in this case N199 MCDB. Using sterile scalpel blade and forceps mince the tissue so that single cells will be come out. Then collect all the media containing separated cells into 15 ml falcon tube and centrifuge it. After that, we will form the cell pellet at the bottom of the tube. Discard all the supernatant and add fresh media into it. Then transfer the aliquots along with fresh media to 6-well plate. Clusters of the ovarian surface epithelial cells can be visualized floating in the medium in each of the well. Erythrocytes will likely be present but usually in very small amounts and they will be removed with the first medium change. Then you just put it in a CO2 incubator for their growth. Leave the plate of the cells in the incubator for at least 3 to 4 days prior to the first medium replacement. These are the real images of ovarian surface epithelial cells, courtesy of course Google Images. In this figure 1, we can see the small clusters of ovarian surface epithelial cells freshly isolated from an ovarian tissue sample. Several erythrocytes are also visible in arrowhead. In figure 2, a colony of ovarian surface epithelial cells have attached and started to spread on the tissue culture plate after 4 days. In figure 3, outgrowth of ovarian surface epithelial cells from a cluster has been seen. In figure 4, confluent monolayer of ovarian surface epithelial cells has been seen, which depicts the typical epithelial cobblestone morphology. Now come to the how to grow and culture established cell lines. Here I am taking an example of human kidney proximal tubular epithelial cell line, which is HK2 cells, which is a transformed continuous cell line, and it is a very good model for renal toxicity study. You can purchase many established cell lines from different companies like ATCC, Cadex Bio, etc. NCSS is an institute in Pune who provides cell line with a minimal price. You can check out the link below. So once you order, mostly they will deliver you in frozen condition packed with dry ice. To ensure the highest level of viability, thaw the vial and initiate the culture as soon as possible upon receipt. For initiation the process, thaw the vial by gentle agitation in a 37 degrees Celsius water bath. Keep the o-ring and cap out of the water to reduce the possibility of contamination. Thawing should be rapid. As soon as the contents are thawed, remove the vial from the water bath and decontaminate by dipping or by spraying with 70% ethanol. Then transfer the entire cell suspension from the cryo vial to centrifuge tube containing recommended volume of specific complete media and spin it for 5 minutes. I'll give you the details of all the media in my upcoming video. Check the viability of the cells and then plate it on flask or culture plates and then incubate the culture at 37 degrees Celsius in a suitable incubator or CO2 incubator. So these are the real images of 6-day culturing cells. Cuboidal and cobblestone morphology appeared. Right side image showed 100% confluency and ready to subculture. So finally, before designing the project, you should keep in mind some criteria for selecting the appropriate cell line for research. At which species you need to work, what are the functional characteristics they have, should be finite or infinite, normal or transformed, what are the growth conditions and characteristics they have, and other criteria also. So thank you for listening. For upcoming videos, don't forget to subscribe and give thumbs up if you like this video. Stay tuned. Take care.